Hello, everyone, and welcome to the Fundamentals of Enteric Neuroscience and Neurogastroenterology webinar series presented by the American Neurogastroenterology and Motility Society. I'm your host, Brian Gulbranson, and I'm a professor of physiology at Michigan State University. And as many of you know, the goal of this series is really to highlight cutting-edge research in gut neuroscience from across the field. And tonight, we really have a great example of that, highlighting 4 young investigators who are doing some of the best and brightest work across enteric neuroscience. So these young investigators come from what's called the Young Investigator Forum. That's a forum arranged by the American Neurogastroenterology and Motility Society to foster growth and development of upcoming young investigators in the field. And we hope that this session is going to be the first in a yearly tradition where we highlight up-and-coming young investigators in enteric neuroscience that come from either the Young Investigator Forum or the Little Brain, Big Brain Forum, both supported by the American Neurogastroenterology and Motility Society. So tonight, we also have 2 outstanding hosts that will be our moderators. So Kyle Stoller and Dr. Yulia Ganz were chairs of the Young Investigator Forum this year. And Dr. Stoller is a gastroenterologist and the director of the Gastrointestinal Motility Laboratory at Mass General. And he's also an assistant professor of medicine at Harvard Medical School and a member of the Clinical and Translational Epidemiology Unit. So Dr. Stoller received his medical degree from Harvard Medical School and trained at Mass General Hospital for residency and his GI fellowship. Dr. Stoller specializes in disorders of gastrointestinal motility and disorders of gut-brain interaction, and his research interests include clinical and epidemiological research in neurogastroenterology and motility, with particular interest in chronic constipation, irritable bowel syndrome, fecal incontinence, eating disorders, and women's health and functional GI diseases. And his research is well funded by the National Institutes of Health. Dr. Yulia Ganz is an associate professor in the Department of Integrative Biology at Michigan State University. She is a developmental biologist who uses zebrafish as her preferred research organism. During her career, she is focused on the nervous system spanning from brain to gut, and Dr. Ganz joined the Department of Integrative Biology at MSU in 2016. And since that time, she has been researching the genes that regulate the formation of the enteric nervous system and studying what this tells us about diseases that affect our gut and digestion. Dr. Ganz has contributed to over 25 publications and won prestigious awards, including an NSF Career Award. So with that, I'll turn the session over to you and you two, and look forward to seeing the great talks.
Thank you, Brian. It's really my pleasure to introduce the first of our young investigators, and that's Dr. Naomi Tjaden from Children's Hospital of Philadelphia. Dr. Tjaden completed her MD PhD training at the University of Kansas Medical Center and Stower's Institute for Medical Research, and continued her training at the Baylor College of Medicine Pediatrician Science Training Program. She recently completed her pediatric gastroenterology fellowship at the Children's Hospital of Philadelphia and stayed on faculty, where she's mentored by Dr. Robert Horkeroth. They're working to define the cellular and molecular mechanisms that affect survival in Hirschsprung's associated enterocolitis, as well as identifying dietary and microbial components that may alter the risk in that disease. Dr. Tjaden, I'll turn it over to you.
Thank you so much, Kyle. Can you guys see my screen?
Yes, good.
That's a yes. Okay, great. So thanks a lot for the invitation. I'm really pleased to be giving my science to this group. It's been a really influential membership to have, and so thank you guys so much. So as Kyle said, I'm going to be talking about how we are trying to modulate microbial dysbiosis as a novel treatment for Hirschsprung-associated enterocolitis. So as a super brief introduction, we study Hirschsprung disease and Hirschsprung-associated enterocolitis. And Hirschsprung disease is when you have the congenital absence of nerves in the colon. This is, let me see, which is shown here. So the enteric neurons we think about as controlling bowel peristalsis, but they're really important for coordinating immune function and regulating epithelial health. And while surgery is provided to these children and it's often life-saving, there are still Hirschsprung disease complications that can persist. And the one that I want to talk about today is Hirschsprung-associated enterocolitis. So you can see in this little cartoon, the normal bowel here on the left and the Hirschsprung-associated enterocolitis here on the right. So on the left side, you can see that the bowel is sort of at peace with the microbes on the luminal side, a nice mucosal layer that's protecting the epithelium that's fully intact, and the enteric nervous system kind of in the middle, sort of from my perspective, orchestrating all of this. And on the right, you have Hirschsprung-associated enterocolitis, where you have a lot more bacteria, dysbiosis, an abnormal mucus layer, and then this epithelial permeability and bacterial entry that occurs. And that's sort of met with this inadequate immune response. And we think that this is what's leading to the sepsis and subsequent death. Now, despite us knowing that, there's only been one really big study that looks at how we can actually change Hirschsprung-associated enterocolitis by using diet. And that was this really nice study done by Tang et al. In 2020, sort of built off this idea that children with Hirschsprung disease and Hirschsprung-associated enterocolitis have this abnormal microbiome composition, particularly with this increase in Enterobacteriaceae. You may recognize Enterobacteriaceae as being implicated in things like inflammatory bowel disease or obesity, but it's also been found to be increased in Hirschsprung disease. And what the study did was they took human children and they looked at kids who were either exclusively breastfed or not exclusively breastfed, and found that those on the exclusive breastfed diet have a 40% less likely chance to get Hirschsprung-associated enterocolitis.
Dr. Tjaden, sorry to interrupt. Just if you could switch to display view, you're in presenter view right now.
Oh, so sorry about that. They attribute this to modulating the gut microbiome, and what you can see is they found a decreased amount of Enterobacteriaceae in the children who are exclusively breastfed, and this increase in these different bacteria that increase the amount of short-chain fatty acids that are in the luminal bowel. Now, we are trying to study a similar thing using EDNRB or piebald lethal mice that we're using as a model for Waardenburg-Shaw syndrome type 4. You can see that the mice and humans have a lot of likenesses, including aganglionic megacolon or Hirschsprung disease, sensory neural deafness, and a melanocyte defect, makes it convenient to know who has Hirschsprung disease in the mice. And unlike the humans who are dying of the Hirschsprung-associated enterocolitis, the mice are actually dying of sepsis. I'm sorry, I meant to say like the humans. And so we know that these mice are dying of sepsis, which makes them a really nice model for what we're looking at. So this all sort of started after a serendipitous finding that location changed the survival in piebald lethal mice. So we were studying these mice here at CHOP in Pennsylvania and knew that the mice were living this very long time point. And when we sent the mice over to Canada for a collaboration, we found that they died really precipitously. And so when you look at the survival curve, again, this green color is in CHOP, the piebald lethal mice, and when they go to Canada, they died quite quickly. We wanted to figure out what was going on and why this could be happening. And so we looked at quite a few different things. One of the first things that we looked at was the degree of aganglionosis, because again, these are Hirschsprung disease mice, and we wanted to see if the degree of how far up the bowel that there was no neurons was actually changed in these two different populations. And what we see is that there are no differences there. And what you can see is, and you'll see this throughout the rest of the talk, is that the mice were housed at UQAM, or the University of Quebec at Montreal, and they were fed the same food, and it's the SLSL, or the piebald lethal mice, versus the mice here housed at CHOP on the CHOP diet. And we also looked at the microbes by doing 16S sequencing, and what you can find is that the microbes actually segregate out really nicely based on the location and actually their diet as well, and not so much by whether or not they have Hirschsprung or if they don't have Hirschsprung disease. When we did an untargeted metabolomic analysis of these mice, what we found is that the food, although both of these are considered completely standard rodent chows, they are quite different in their components at CHOP versus UQAM for, again, our standard chows. And when we look for different aspects in the stool using this untargeted metabolomics and picking out things that we had a particular interest in, such as short-chain fatty acids, we do see that we have this increased amount of stool butyrate while the mice are on this CHOP diet. So what we've done now is we've done a diet swap at both institutions. So first we'll look at CHOP. So the CHOP piebald lethal mice on the CHOP diet, again, shown here in green, live for a very long time. When they get swapped to the UQAM diet, you can see that they die much quicker, both on the first, or the F1, and the second, or the F2 generations. When we do the same thing at Canada, or UQAM, you see again the Canadian piebald lethal mice on the Canadian diet die quite quickly. You put them on the CHOP diet and they live for a long time, and this is actually increased by the generations that they're on. So on the second generation of being on the CHOP diet, they live even longer. And because of this finding, we've done now the rest of our experiments at the F2 or the second generation and onwards, which is what I'll show you now. So because we knew the human data of the increased enterobacteriaceae in Hirschsprung and Hirschsprung-associated enterocolitis, we also looked at this in the mice. And these are all mice housed at CHOP, either on the UCAM diet or the CHOP diet. And what we have are the piebald lethal mice as compared to their SS litter mates, which are not exactly wild type mice, they are hypomorphs. And what you can see is that the piebald lethal mice have more enterobacteriaceae or this dysbiotic bacteria. And then when they're on the UCAM diet, it's increased even more. When we look at true wild type mice or C57 black 6 mice, and we do this for multiple generations, we again see you have a much increased amount of enterobacteriaceae while on the UCAM diet. So we wanted to know if inhibiting this would actually change your survival. So we used a medicine called sodium tungstate that was actually shown in a DSS colitis model to decrease the amount of enterobacteriaceae in the setting of inflammation. And what we did is that we administered tungstate in the drinking water in the second generation or beyond, right at the time of weaning for about 1 week. And what we found is that, again, this is the same survival curve that you've seen before, that when the mice are on the UCAM diet on regular water, they die quite quickly. But when we provide about 1 week of tungstate therapy, you see that their survival has increased quite a lot. And this was just a pilot study that we actually ended because the mice were living for so long, so we could repeat the study. And to show you what we think is happening is that you can see these mice at the beginning of their tungstate therapy on day 0 of therapy, they have quite a high enterobacteriaceae burden. And the longer they're on this therapy, we see that that decreases quite significantly. And so looking at what mechanisms we think might be driving this diet-driven survival, we also did a large RNA-seq experiment where we took mice who are housed at CHOP and are either on the CHOP in green or UCAM diet. We looked at both the proximal ganglionated colon as well as the distal aganglionic colon. And then we took 1.5 centimeters and then we did a bulk epithelial RNA sequencing experiment. I'll just show a little bit of that data today. So what you see, again, with the PCA08 plot, you can see that the distal colon, the proximal colon samples segregate out really nicely, which is not unexpected. But what's really cool to us is that in the proximal colon samples, you can see that the two diets actually do split out much nicer than in the distal colon. And when we look at the GO-term analyses of what is actually being changed, and this is just in the piebald lethal mice, but we did do this with their SS litter mates as well. But just to draw your attention here to the mice here on this top part, you can see these are the gene changes that were much higher when on the CHOP diet, at least 1.5-fold difference with a p-value of 0.05. And you can see the things that are high in the proximal colon versus distal colon, and then the high expressing genes that are in the, highly expressed while in the UCAM diet. And I just want to zoom in a little bit here for time to look at the proximal colon, and a couple of things really come to our attention. One is the fucose catabolism processes, and 2 is the antimicrobial peptides. And the reason we're so interested in these is because previous scientists have shown that VIP-specific enteric neurons can direct ILC expression of IL-22 to increase these genes, the fucosylation and antimicrobial peptide genes, that are actually protective against these detrimental microbes such as proteobacteria, and that they can actually improve the amount of butyrogenic microbes that are able to grow, which then go into this positive feedback loop where we're increasing the amount of butyrate, which is used as an energy source for your colonocytes, that go into this positive cycle of, again, increasing fucosylation and antimicrobial peptides, improving your tight junction and as well as increasing more IL-22 production. And what we found is that the CHOP diet plays into this by increasing stool butyrate, like we saw at the beginning, decreasing the amount of the detrimental bacteria and increasing fucosylation and antimicrobial peptides. So if you'd like to hear more, I'm happy to ask more questions or answer more questions at the end, and we can talk about some of the future directions. But with that, I'd like to end here with acknowledgements, thanking, of course, our fearless leader, Bob Quakeroat, the rest of the lab, and especially the people here in Hot Pink who've done a lot of this work, along with our collaborators, ANMS for the invite, and then, of course, CHOP and our funding sources.
Dr. Tjaden, thank you so much for that fantastic presentation. Just as a reminder, for those of you who are tuning in, there is a Q&A box at the bottom of your Zoom window, and please send questions for any of the presentations, and we will have the panelists on afterward to answer those questions in real time. So now we're going to transition over to more of a clinical research focus. And really my pleasure to introduce another one of our young investigators, and that's Dr. Yuying Luo from Mount Sinai West in Morningside in New York City. Dr. Luo is an assistant professor of medicine at Mount Sinai West in Morningside, where she's also part of the Mount Sinai Center for Gastroenterology and Physiology. Her clinical and research interests include disorders of gut-brain interaction, lower gastrointestinal motility, and women's GI health. Now I'll turn it over to Dr. Luo. Thank you.
Thank you, Dr. Stoller. Thank you to the ANMS organizers and moderators for inviting me to share some of our preliminary data on IBS in the pregnant and postpartum population. IBS, or irritable bowel syndrome, is one of the most prevalent disorder of gut-brain interaction globally. Studies show that it affects about 5 to 10% of the general adult American population, and like other DGBIs, impacts women at a ratio of about 2 times that of men. Besides prevalence, there are sex-based differences in terms of IBS symptomatology. A meta-analysis of 20 studies evaluating gender differences in IBS found that women were more likely to report constipation-related and abdominal pain symptoms compared to men. In the same study, the authors also showed that both IBS and healthy women reported increased frequency and severity of IBS-like symptoms during menstruation, suggesting a hormonal component. In addition, women with IBS have a higher prevalence of overlap with other chronic pain disorders, such as fibromyalgia, chronic pelvic pain, and migraines. Perhaps it is not surprising then that women with IBS are found to have a lower quality of life, with more fatigue, depressed mood, and anxiety compared to that of men. A large proportion of women affected with IBS are of childbearing age. However, there's a paucity of studies that specifically address the epidemiology and management of IBS in the pregnancy and postpartum period. This is also the population that has largely been excluded from clinical trials. This figure is from a recent review on IBS in pregnancy, highlighting the many physiologic changes which can impact previously existing or lead to the development of new onset IBS. For example, estrogen can affect both peripheral and central regulatory mechanisms, leading to increased visceral hypersensitivity. Aside from physiologic factors, we all know psychosocial factors also play a role in the development of IBS. This is a schematic from an older review highlighting the other important modulating factors that impact the development and severity of IBS in women, including biobehavioral stress responses, the gender role, and psychological symptoms, all of which undergo significant fluctuations during this pregnancy and postpartum period. The aims of this study were twofold: to determine the prevalence of IBS in pregnant and postpartum women, and to characterize the clinical profile of this population. We conducted a cross-sectional claims analysis using a national de-identified database, IBM Health Market Scan. Utilizing previous validated methodologies, we created a cohort of all delivery hospitalizations resulting in a live pregnancy for individuals 15 to 49 years of age between the years of 2013 to 2019, and merged with linked outpatient claims. Patients with only an IBS diagnosis as an inpatient and no outpatient claims were excluded, as well as those with diagnoses that were similar to IBS, such as celiac, GI cancers, and inflammatory bowel disease. We collected demographic, obstetric, as outlined by the CDC Severe Maternal Comorbidity Index, psychological, and other medical comorbidities, including connective and autoimmune diseases, based on previous studies highlighting the comorbid nature of these diseases and IBS. We were able to create a cohort of 1.6 million deliveries with regional representation across the US, which was proportional to the census population. The prevalence of IBS in our cohort was 1.76%, with 0.8% of our cohort having a new IBS-related claim after delivery, with the majority of these claims occurring in the first year postpartum. In terms of looking at the subtypes of IBS, IBS unspecified was the most common. For the years where ICD-9 coding was used, there was no IBS subtypes available. IBS unspecified was followed by IBS diarrhea as the second most prevalent, followed then by IBS constipation and mixed type of IBS. For patients who did not have an IBS-related claim prior to the time of pregnancy, we also calculated the overall incidence density, which was 52.7 per 10,000 person years. The average mean follow-up of patients in our study was about just under 2 years. Amongst IBS subtypes, the IBS unspecified also had the highest incidence density. We also looked at severe obstetric comorbidities, and although we observed some statistically significant differences between the groups in conditions such as acute renal failure, adult respiratory distress syndrome, eclampsia, and disseminated intravascular coagulation, these were deemed not to be ultimately clinically significant given the overall low number of these cases. And for the main finding of our studies, we found that pregnant and postpartum patients with IBS were more likely to have psychological comorbidities, such as depression, postpartum depression, and anxiety. They were also more likely to also have comorbid migraines and connective tissue disease and autoimmune diseases, although the overall number of cases for the latter was much lower compared to other comorbidities. A higher BMI of over 40 was also more prevalent in pregnant and postpartum patients with IBS. So to summarize, we found that the prevalence of IBS in our pregnant postpartum cohort was about 1.76%. This is much lower compared to the prevalence in the general adult population. Reasons this might be include lower healthcare utilization for pre-existing IBS during the pregnancy and postpartum period, or reflect that IBS severity may actually improve. Of the new IBS claims postpartum, the majority occurred in the first year. This is the mean follow-up in our study for postpartum women who did not have an IBS claim prior to pregnancy was just under 2 years, and may also reflect that these are the patients who have severe enough symptoms to seek healthcare during this time period. Importantly, we identified that in pregnant and postpartum women with IBS, there was a higher prevalence of psychological comorbidities such as anxiety, depression, postpartum depression, migraines, connective tissue, and autoimmune diseases. While 3/4 of women may report some functional bowel symptoms during pregnancy, our findings may inform a targeted population to screen for and monitor for the development or worsening of chronic bowel symptoms, which may then meet the criteria for IBS, necessitating interdisciplinary care. These comorbidities, as well as IBS, can impair health-related quality of life and maternal well-being. As for the limitations, there are always inherent limitations to using claims-based databases to assess for prevalence of IBS, as patients may not utilize healthcare services for their symptoms. Other limitations of this particular database include lack of demographic data, such as race, enrichment in commercially insured individuals, which impacts its generalizability. And now the strength of the study include a large number of pregnant women with regional representation proportional to the US Census population and the inclusion of the postpartum period. Our next steps include the exploratory modeling of risk factors, specifically looking at new onset IBS after pregnancy. So in conclusion, psychological comorbidities are prevalent in pregnant and postpartum women with IBS. And future studies are needed to validate specific risk factors for increased IBS severity or the development of new onset IBS in pregnant and postpartum patients. The potential comorbidities that we highlighted may identify a specific population where increased attention may be needed to manage existing IBS and to monitor for the new development of new onset IBS. Thank you.
Thanks so much. So our next speaker is Dr. Amy Shepherd at Boston Children's Hospital. So Dr. Amy Shepherd is a postdoctoral researcher fellow in Dr. Mingtashi Rao's lab at Boston Children's Hospital, where research focuses on how enteric nervous system and epithelial signaling can modulate GI function. Prior to this, she was a tech researching mucosal immunology in New Zealand, and then went on to complete her PhD in behavioral neuroscience at the University of Melbourne in Australia. In her future, she's interested in combining her expertise in immunology and behavioral neuroscience in the context of neuroepithelial signaling in the gut. Looking forward to your talk.
Thank you so much for the introduction, Yulia. And hi, everyone. Thanks for coming tonight. So today I'm really excited to talk to you about some work that's just very recently been published and where we're
Sorry
going
to
here.
interrupt.
Yes.
Can you set it to the presenter mode?
Like this, does that look better?
Perfect, thank you.
Perfect, great. Thank you so much. So this audience already knows a lot about RET signaling, especially in the development of the enteric nervous system, where RET positive enteric nervous system precursors move down a GDF gradient to what will become the terminal end of the colon. And when this process is interrupted, you can develop Hirschsprung's disease that was introduced really beautifully by Naomi earlier this evening. And this affects about 1 to 5,000 patients and has a really striking sexual dimorphism where it affects 4 to 1 male to female patients. Treatment is surgical resection of the aganglionic bowel, but post-operative Hirschsprung's disease patients commonly present after these surgeries with a high risk of enterocolitis, as well as chronic defecatory dysfunction. And these two symptoms or kind of observations led us to think that RET signaling may not only be important in the development of the enteric nervous system, but also in the normal function. This has been, we found a little bit of evidence for this as well with recently published RET kinase inhibitors that are past FDA trials for cancers with high RET expression, and patients on these drugs have very high rates of both constipation and diarrhea. And so this led even more credence to the idea that RET signaling in an adult intestine is important for normal function. So where is RET in the normal intestine? Previous studies had already shown that small innate lymphoid cell populations express RET in the intestine, along with enteric neurons, unsurprisingly, and a rare cell population up around the epithelium called enteroendocrine cells. But while we knew it was expressed in neurons and enteroendocrine cells, the extent of expression and in which subtypes was unknown before we started this work. So first to the neurons, what we found when we wanted to quantify the extent of expression in 4, 12, and 52 weeks age-old animals, we found about 50% of all neurons in the myenteric plexus of the ileum and the colon expressed RET, and about 70% of all neurons continue to express it in the submucosal plexus of the colon, so a really large proportion of enteric neurons. When we looked at subsets, while we found that some calbindin-1 neurons expressed RET, we thought it was really interesting that all inhibitory motor neurons, as characterized by NOS1 expression, shown in this image here, expressed RET as well, both in the ileum and in the colon. And so we're really interested to see if perturbing RET expression in these adult populations would alter either neuronal survival or GI motility. To do this, we utilized a mouse model that would have normal RET expression and development, but one that we could take out RET in adulthood. We're crossing a RET CRE-ER with a RET flox EGFP. When these mice are induced with tamoxifen, it will cause the excisation of RET from the normal locus, and then the expression of this EGFP protein, allowing us to visualize the cells that have lost RET in a longitudinal fashion. And so we induced these mice in adulthood and looked 1 month later at their enteric nervous system, and we saw that it looked pretty normal with a very high expression of GFP, indicating that these neurons are RET deficient and living and looking pretty normal. And you can see high overlap with NOS1 and with UCD. And when we quantified this compared to their wild type counterparts, which have no GFP expression, we saw the same proportion of neurons both in the ileum and the colon myenteric plexus, indicating that RET signaling, at least in this adult stage, is not important for neuronal survival. And this is in contrast to previous studies that look early postnatally or in development, in which case it really is very much required. So we then wanted to look to see if RET expression was important for modulating GI motility. So we took our RET conditional knockout mice, we looked at their GI motility, compared their GI motility to their haploinsufficient counterparts, and found that 2 weeks after RET deletion, we see about a 30% slowing in GI motility that really interestingly had started to normalize by 4 weeks post-induction. Now I just showed you that the neurons are still there 4 weeks post-induction. And so we went back to the drawing board and started to think about these enteroendocrine cells in the epithelium and wondered if maybe they were the cause of this transient slowing in GI motility, because the turnover of enteroendocrine cells is very high, which means that we'd be getting a lot more RET sufficient enteroendocrine cells by this 4-week time point. So to look at this first, we went back to our RET CRE-ER mouse and crossed it now with a Rosa TD tomato and followed longitudinally to see if we could see the expression of RET positive enteroendocrine cells. And indeed, what we saw both at 1 and 2 weeks post-induction is we could see the presence of these RET positive red cells in the epithelium. But when we were looking at 3 and even 4 weeks post-induction, we found very few, if any, RET positive enteroendocrine cells, leading us to hypothesize that the slowing in GI motility was in fact due to enteroendocrine cells, RET signaling on enteroendocrine cells rather than enteric neurons. To test this further, we generated a RET epithelial specific knockout mouse by crossing a Villain-Cree with the RET flox mouse. When we looked at GI motility in these animals, we saw a similar effect where of about 30% slowing in RET epithelial knockout males. But interestingly, we didn't see the same effect in females. So it seems to be the males are much more vulnerable to having deficient RET signaling, mirroring what we see in Hirschsprung's disease patients. We then wanted to test this with a pharmacological measure. So we utilized a GI-restricted RET kinase inhibitor called GSK408B and administered this to mice for 10 days, 10 milligrams per kilogram, and then assessed GI motility. And this exactly phenocopied what we had seen in the RET epithelial knockout mice, with about a 30% slowing in GI motility in males, but not female RET kinase inhibitor treated mice. And further studies that I'm not going to show you today indicated that this slowing was really due to slowing in the small intestine, not to slowing of gastric emptying, nor changes in colonic motility. So we then wondered how many enteroendocrine cells and what type were expressing RET. So we first looked at chromogranin A to look at mature enteroendocrine cells and found that, like enteric neurons, about 50% of all enteroendocrine cells express RET. So we're talking about a really significant population here. And interestingly, we found that a very large amount of L cells, enteroendocrine cell subset, expressed RET as well, especially in the distal small intestine. And L cells are hypothesized to be involved in a phenomenon called the ileal break, where when nutrients reach the distal small intestine, where they are very concentrated and the nutrients are sensed there, these cells will release GLP-1 and PYY, which then signal to the intrinsic and extrinsic nervous system to slow small intestinal motility and suppress appetite to allow those nutrients to be fully absorbed. And if this is true, that would make sense that what it means is when you have deficient RET signaling, you're going to have exaggerated PYY and GLP-1 release, which is then going to slow gut motility. So this is our kind of working model at this point. And so to test to see if our model was correct, the first thing we wanted to see is if we block RET signaling in enteroendocrine cells, do we see exaggerated PYY and GLP-1 release? To do this, we went back to our drug model and took serum from fasted or 2-hour postprandial drug or vehicle treated male and female mice. So first of all, looking at fasting, as expected, levels of GLP-1 and PYY were very low in males and females and in vehicle drug treated mice, exactly what we would expect from a fasted state. When we fed the females, we see a really nice peak in both GLP-1 and PYY in a way that is unaffected by drug, consistent with the prior observation that females don't have any changes in GI motility. When we look at our males, interestingly, we see almost like a failure of the fed mice to have a peak at 2 hours, indicating that RET kinase inhibition is either altering the amount of GLP-1 and PYY release or the kinetics of that release compared to their vehicle-treated counterparts in males. And so with this evidence, we went through and thought a little bit more about this model and thought that if this model was to be true, and the fact that because of the fact we had seen GLP-1 and PYY were unchanged during fasting, we wanted to see if the GI motility slowing that we see in the RET epithelial knockout mice would be abrogated in a fasting state. So we did this, and that's exactly what we saw, where that 30% slowing is rescued by fasting the mice prior to starting GITT. And the other thing that should happen is if we block the PYY receptor, NPY2R, or the GLP-1 receptor, GLP-1, that should rescue this motility effect. And indeed, that's what we saw when we treated the mice with a PYY receptor antagonist. It isn't quite the same case for the GLP-1 receptor antagonist, which either could be, but unfortunately that drug has a very, very fast half-life. So I'm not sure if that means that blocking GLP-1 receptor is not sufficient, or if it was just a kinetics problem. Finally, we wanted to check that this was the same in both human enteroendocrine cells as well as mouse. So we utilized human intestinal organoids with David Brault's lab here at Children's, and treated these enteroendocrine cell containing intestinal epithelial organoids with an FDA-approved RET kinase inhibitor, selpercatinib. And what we found is with increasing doses of selpercatinib, we get increased release of GLP-1. And so this is, as I said, this work has been very recently published. And so what's next? First of all, I'm really interested, I've been talking about RET signaling like it's one thing, but there are actually multiple RET ligands. The most famous is the glial cell line-derived neurotrophic factor, GDNF, but there are other members of this family, nurturin, artemin, and perspirin, all of which signal through RET with their respective GFR alpha receptors. And we don't know which one of these ligands is driving this effect in enteroendocrine cells, and if there may be, in fact, different effects with different RET ligands. We also don't know the main source of the RET ligands that are signaling to the enteroendocrine cells. There are a variety of cell populations that can make different combinations of these RET ligands, and we're really interested to see how each of these populations may be working with enteroendocrine cells to maintain normal motility. And finally, I'm really interested in the role of RET signaling in EECs and inflammation in the context of enterocolitis, like Naomi was talking about, and seeing if that may help promote barrier function, and if it could explain why some of these Hirschsprung's disease patients are really vulnerable to enterocolitis. And with that, I'll thank you all for your attention. I'll be thrilled to answer questions in the question time. Thank you so much.
Thank you, Amy. So our final speaker will be Dr. Jacques Gonzales. Dr. Gonzales holds a Master of Science in Neuroscience from the University of Strasbourg and a PhD in Neurogastroenterology from the University of Nantes in France. He completed his PhD under the guidance of Dr. Michael Neuenlist, where he studied the relationship between gut microbiota and the intestinal dysfunction observed in patients with autism spectrum disorder. During his master and PhD studies, Jacques developed a keen interest in glial cells and decided to join Dr. Brian Gulbranson's lab for his postdoctoral training at Michigan State University in 2021. His research is currently focused on the remodeling of the enteric nervous system after early life adversity and the role of enteric glia in inflammatory GI disease. Take it away, Jacques.
Thank you for the introduction, and thank you, ANMS, to invite me tonight to present at this webinar series. So, as you all probably know, DGBI is a widespread GI condition which affects more than 40% of the world population, with a higher prevalence in women than men. This condition induces change in neuroplasticity that leads to GI dysfunction, profoundly affecting the overall quality of life of the patient. Despite the high prevalence of DGBI, the exact factors causing this condition are not completely understood yet. Some risk factors, such as the biological sex, inflammation, or stress, has emerged as a crucial contributor to the onset of this condition. However, the study of DGBI is becoming increasingly complex as these risk factors interact with each other, amplifying the susceptibility to the pathology. For instance, while DGBI are more prevalent in women, an exposure to stress could also increase the predisposition of men to develop DGBI. Among the different stressors the human could be affected by, several studies show on human patients an increase in DGBI onset after the experience of an early life adversity. In fact, the nature, the frequency, and the severity of this stressor correlate with the susceptibility of developing DGBI. And this correlation between early life adversity and gastrointestinal symptoms has been reproduced in animal models. Very interestingly, human and animal studies show that the impact of specific early life stressor could affect differently the biological sexes. For instance, here, a male subject to maternal neglect paradigm show an increase in visceral sensibility, while females are not, in fact, affected by this early life stressor. But yet some crucial questions remain. What makes female more susceptible to develop DGBI, and what triggers switch in male leading to the development of DGBI despite their lower sensibility? The enteric nervous system, the main regulator of intestinal function, is very sensitive to stress exposure during the postnatal period. And among its components, the enteric glial cells are known to play a crucial role in the homeostasis of the intestine. The cells are, in fact, involved in the regulation of pain response, motor function, inflammation, and neuronal communication. Several research on enteric glia demonstrated that alteration of the cells lead to intestinal dysfunction manifested by DGBI-related symptoms. So all of this lead us to our hypothesis, which was how early life adversity affects glia, and are they essential actors in the onset of DGBI after early life adversity? So we in the first step, we decide to study the gene expression of enteric glia and how this gene expression could be affected by early life adversity. To do so, we use a ribotaxoxem animal, which allow to insert a specific tag on the SOX10 positive cells, so the glial cells, and will help us to purify the mRNA from enteric glia. For this study, the animal was separated in two groups, one control group, which is called a normal enlid, and one ANMS group, where the pups were subject to a daily separation of 3 hours and combined with an early weaning at postnatal day 70. During the adulthood, we collected the intestinal sample, extract the specific mRNA from glial cells, and realize a bulk RNA sequencing. So first, we decide to observe the difference in baseline between male and female enteric glial cells. And we observe is male have different genic expression profiles than females. As you can observe on this volcano plot, female has more than 400 genes differentially expressed in comparison to male, and most of them trend to have a higher expression. And when we focus on the different pathway related to this gene, we can observe that female have a higher expression of gene related to gastrointestinal disease or inflammatory disease. And when we focus on the canonical pathway affected by this gene, we can observe that there is a change in the expression of GPCR signaling or calcium signaling, which are known to be very important for glial function. In a second step, we decide to focus on the difference induced by early life adversity on enteric glia. So on the left, you can observe that after experiencing early life adversity, male enteric glia have a huge shift in their genic expression with a high increase of most of more or less 600 genes. While on the opposite, in female, you can observe a lower impact with mainly a decrease in gene expression. So this result indicates that early life adversity affects both sexes, but at a different proportion. And the very interesting thing in our bulk RNA sequencing is when we focus on the gene difference between male and female enteric glia in baseline, and we compare them with the one differentially expressed in male after NMS, we observed an important overlap in this gene differentially expressed, suggesting that male enteric glia is getting closer to the female gene signature, inducing so a removal of enteric glia's sexual dimorphism. So, as I presented earlier, enteric glia is sexual dimorphic cells with differential gene expression in signaling pathways such as calcium activity known to be very important for glial function. So does early life adversity alter the enteric glial communication? So to assess this, we use a transgenic animal expressing a GCAMP indicator in the enteric nervous system cells, coupled with the expression of a DREAD receptor on GFAP positive cells. And this transgenic animal allows us to directly stimulate enteric glia with CNO and record directly the activity response to this stimulation. So here is an example of a reaction to CNO stimulation. So you can observe a huge wave of calcium response after the CNO stimulation and a comeback to baseline. So here you can see some representation trace from male in the green box and from female in the yellow box. And visually, you can observe directly that the trace between male and female are totally different. I did a quantification of the difference, and I observed that the calcium activity in enteric glia is significantly reduced in male in comparison to female. Then in the next step, we decide to focus on how early life adversity altered this glial communication. So on the left, still the same video as I show you with the first initial wave of calcium. And on the right, this time you can say a wave of calcium, but if you wait a little bit, you can start to observe a lot of post-activity after the initial stimulation. So this video were transcribed in trace again. On the top, this is the previous trace I showed you before, and in blue there, that's the trace after experiencing early life adversity. On the left, male, on the right, female. And here again, you can see a clear alteration of the calcium response after experiencing early life adversity, characterized by a decrease of the initial response and a modification of the post-stimulation activity. So we realize a quantification of these different traces and find that early life adversity reduced the calcium activity in both sexes, and more interestingly, it decreased the sexual difference between male and female. So as I told you before, calcium activity is very important in glial function. So is there any functional consequences due to the observed change in enteric glia? We decide to focus on intestinal motility, which is known to be strongly regulated by enteric glia. We use a common red common gavage, and we inject the animal with CNO to specifically activate enteric glia. So here is the result. On the left, this is a control animal. In blue, the stress animal. And each of these treatments are separated with animal not expressing the dread receptor and animal expressing the dread receptor. So first, you can observe that between control animal and early life adversity animal, there is a delay more or less by twice of the gastrointestinal transit time. And when we stimulate specifically enteroglial cells, there is a reduce of the difference between control and early life adversity, suggesting that enteroglia could be partially responsible of the GI dysmotility observed. And the last thing I want to show you is that when we split with the animal by sex, we observe a very interesting result. So in both sexes, there is a delay in the intestinal motility, but only in males, the specific glial activation restores the gastrointestinal transit time to a baseline. So in conclusion today, I show you that enteroglia are sexually dimorphic cells with difference in gene expression and a variety of calcium communication. And early life adversity eliminate glial sexual dimorphism by affecting directly the gene expression and the calcium communication. And this early life adversity increase gastrointestinal transit time, partially due to the alteration in enteroglia. And to conclude, enteroglia are a major actor in gastrointestinal disorder induced by early life adversity and could be one of the explanations of the sexual dimorphism observed in DGBI. I would like to thank all of my lab and every collaborator for this project, and thank you for your attention.
Well, thank you to all of our speakers tonight. These have been some fantastic talks. And just a reminder for anyone who's in the audience is that you can use the Q&A function at the bottom of your Zoom window to ask any questions, and we'll try and relay them live in about the 10 minutes that we have left here. So I'm gonna go in the order of the talks here from the audience. So our first question is for Dr. Tjaden. Dr. Tjaden, great talk. Question, did you look at these in the RET mutant mice in the findings?
No, we haven't. That's a great idea. We've actually been thinking about doing this in other models, I think I'm going to need Amy Shepard to help with some of this stuff. But yeah, I think it's a really good idea to make sure that this recapitulates in other gene changes in Hirschsprung disease. And I think it will also give us a little bit more information as to what in the developmental process is so important about the enteric nervous system itself in responding to the different diets and dysbiosis. But great question.
So I have a talk for Amy. So Amy Shepard, great talk. Did you check if there's any cellular changes, either neurons, glia, or enteroendocrine, either in the number or expression of specific markers in the colon, large intestine of the adult rat knockout?
Yeah, so we didn't look at enteroendocrine cells because they turn over quite quickly. Most of the tissue samples I collected were at 4 weeks, and I think most of them would have turned over at that point. But in terms of neuronal cell populations, we see equal amounts of calbindin positive and NOS positive percentage-wise to their wild type counterparts. And in the rat EGFP flox mouse, what we see is we see that the inhibitory motor neurons, pretty much all of them are expressing RET, just like we see in the RET reporters that are normal. So I think the neurons populations are pretty much the same. I couldn't say 100%. We haven't looked at everything, but they seem pretty normal. Yeah.
Next question's for Dr. Luo. Great talk, Dr. Luo. Interesting to see that IBS prevalence was lower than expected during pregnancy. Do you think the immunologic changes that associated with pregnancy may be linked to the findings that you saw?
Yeah, I mean, I think anecdotally, a lot of physicians who care for a lot of patients in this population talk about how their patients disappear during the pregnancy period and then come back later. So I think either along the lines of hormonal changes or, as you highlighted, immune system changes can contribute to the differences in the prevalence that we actually saw.
The next question is for Dr. Gonzales. Great talk. How do you relate your findings to humans?
Oh, that's a very interesting question. How I do relate this? So it could be very interesting to study in human if we can still observe this sexual dimorphism in enteroglia, which is doable on biopsy or surgical resection pieces where we are able to do enteroglia culture. But the very interesting thing could be maybe this pro-inflammatory profile of enteroglial cells in female could be one of the reasons of the highest sensibility to inflammation in women during DGBI. But that's just an exploration, and it needs a lot of further investigation to know better on human.
Next question for Dr. Tjaden. Dr. Tjaden, wonderful talk, super interesting data. I may have missed it, but did you look at colonic motility in the EDNRB mice on the 2 diets and also in the SIP syncytium?
Thanks for the question. That's a really good question. We have not looked specifically at the motility or the syncytium. We have been sort of focusing more on the epithelial barrier because I think it's more of a combination of barrier defect plus the dysbiosis more than the motility itself. But it's a great question. It's something for us to think about for sure.
So a question for Dr. Shepard. Excellent talk. Just to clarify, do the organoids you have used have enteric neurons or just in the enteroendocrine cells?
So they are, it's a great question. I wish we had an enteric neurons in there, but they are intestinal epithelial organoids that were taken from human biopsies and then underwent a series of small molecule incubations to really help differentiate a lot of mature enteroendocrine cells. So it's a mix of intestinal epithelial cell populations.
Dr. Luo, you saw in your pregnancy data that there was a high incidence of what seemed like new IBS after delivery. And you also saw that this seemed to be associated with certain comorbidities like psychiatric disease, as well as connective tissue diseases, migraines. Do you have sort of a proposed mechanism by which pregnancy and these potential comorbidities would work together to create IBS?
That's a great question. I mean, I think this dovetails nicely into some of the basic science talks. We really need more research in this area. I think for me especially, I think clinically, I think it has to do with the hormonal changes and probably also along the lines that these comorbidities run with, central sensitization disorders. So something along the lines, I think a proposed mechanism would be like hormonal fluctuations in patients who may be already predisposed to developing disorders of visceral hypersensitivity and central sensitization.
Question for Dr. Gonzales. Do you think the changes with calcium communication affect ICC signaling?
Oh, that's a good one. Thank you for the question. So the calcium imaging I'm doing is focused mainly on the intraganglionic cells, but probably it will be better to do this kind of study directly on the intramuscular glial cells. But I will probably tell you yes, as this modification affects neuronal communication with the surrounding environment, it probably affects lately ICC.
Great. A question for multiple speakers here of our basic scientists. How are you measuring intestinal permeability or barrier function in the work that you're doing? We'll start with Dr. Tjaden, go in order.
All right, sounds good. We've done it in two different ways. One is with Yuying chambers and the other way is with Fitzy-Dextran assays. The first is tricky. You have to find someone who knows how to do it and has all the setup ready to go. And so we found ourselves having a collaborator here who was doing that with us, and then they've actually moved. So we've actually switched now to the Fitzy-Dextran, which regular old people like me can do.
Dr. Shepard?
Yeah, we also do Fitzy-Dextran, but we have two different sizes that we like to use. We use a 478 Dalton if we're doing kind of like a normal, relatively non-perturbed setting, and then a 4 kilodalton, which you'd use in something like DSS or anything that you would think your epithelial barrier might be perturbed. But yeah, as Naomi says, it's nice because it's a very easy assay to do.
Dr. Gonzales?
I'm doing exactly the same as Amy and Naomi, but we also, in my previous lab, we were studying transcellular permeability, and to do that, we were using very large molecules such as HRP, for example.
Well, I'd like to thank all of the presenters today. We've run out of time with a really engaging set of talks and questions. If you have more questions, this conversation will go online to Doc Matter, as you can see displayed here. And you can answer and interact with some of the speakers that you've heard tonight. Again, thanking all the speakers and especially ANMS for hosting this great event. And we'll see you for the next event.